I'd like to thank you for those remarks and for inviting me. I always also enjoy coming here, and I'm going to give an update on our office, including some things some people may have heard about the reorganization and how it affects the office. And I also would like to echo the mutual respect which we at FDA hold arm. So here's an outline of my talk. So first I'll give an office update. And as was already mentioned by Michael, I'm the Deputy Director now of CBER, the Center for Biologics Evaluation and Research, which is the center which includes the Office of Cell Tissue and Gene Therapy. And we're in the process of actively recruiting for a new office director. So what about the reorganization? I know FDA sent out an announcement and there's something posted on our website. So I thought I would just say a couple words about it as it affects this field. It's effective October 16th. The office will change its name to the Office of Tissues and Advanced Therapies. All of what you currently know as OCT GT will be part of the new office. So from that point of view, there's no splitting up of any of the products that this community is primarily interested in. All of those will be transferred to the new office along with all the employees. So what is new is that some products from the Office of Blood Research and Review will be transferred. Those are, for example, some of the products that treat coagulopathies and some other types of products like snake antivenom that doesn't fit naturally into any specific home except by the products. So the two divisions in OBR that will transfer is two of the divisions, the Division of Hematology Clinical Review. That's going to become part of what you all know as DCEPT. In the next slide, I'll show the new organization. And the Division of Hematology Research and Review, which are the two lab divisions that review the products associated with the products that are transferred, is going to move as a unit. So next slide, I show the office structure. So as you can see, there now are going to be 5 divisions. The Division of Cell and Gene Therapies, that will remain the same. So that's not changing its structure. That's the division that is directed by Dr. Puri. Many of you know Stephen O., Denise Gavin, or Kim Benton. They have the same structure and same people as were there before. The Division of Plasma Protein Therapeutics, those are the 2 lab branches that I mentioned that are going to be transferred, and they do the product reviews for the products that are being transferred. The Division of Clinical Evaluation and Pharm Tox is going to be bigger than it was before, and so it's going to have a new structure of branches. I've listed the branches on these slides so you can see, but basically some of the groups are going to break up into two branches for easier management. The Division of Human Tissues will be unchanged. I do want to mention one thing about the Division of Human Tissues. It doesn't have to do with the reorganization, but we had been recruiting for a new division director since Dr. Lazarus retired last spring, and I'm happy to report that this week Scott Brubaker, whom some of you know as having been the science officer at ATB, has accepted the role of division director for the Division of Human Tissues, so he is now the director of that division. And then the Regulatory Project Management, which was a branch reporting into the Office of the Director, is going to become a division because of the increased number of files. It's expanded. It's going to be 2 branches and will be in a division reporting to the office as such. So will be some of these organizational structures don't have managers in place yet, like that division. So we'll be recruiting that, and there'll be names. So next year when someone comes, hopefully they'll be able to put names in all those slots of who's the director of all the different work units. So I'm going to go back. The rest of these slides are all written from the point of view of OCT GT. The reorganization having been announced fairly recently, I didn't really integrate the new products into my talk, especially given that this audience is primarily interested in the products that were OCT GT products. So I don't want to confuse anyone. These are the products from the the OCT GT that's transferring. So I think a lot of you know these are the kind of products that we review in the office. What I do want to show is that the number of new IND submissions that we have has really been exploding over the last couple of years. I think this reflects a lot of the progress in the field and the enthusiasm in the field that we share also in OCT GT. We share the enthusiasm and the excitement for what's going on in the field. These are new submissions, so this wouldn't tell you how many total studies are active in the office, but it gives you a reflection of the increase in interest over the recent years. I'm going to speak briefly about expedited programs because that's a question everyone's interested in, especially Breakthrough. So Breakthrough is a new program that was signed into law when FDAs was enacted in 2012. And the intent was to expedite development and review of drugs that were intended to treat a serious or life-threatening disease or condition and had preliminary clinical evidence that indicated that the product could have substantial improvement over existing therapies on one or more clinically significant endpoints. There's a guidance, this is just a little, I want to just take a step back and just say there's a guidance that describes all of these expedited programs for serious conditions, and these are the 4 programs it describes. I'm going to be speaking briefly but specifically about Breakthrough. However, this gives you, and this is in the guidance, or I don't know if this table is in, but the information is in the guidance, and it compares the different features of these different programs. So what has been our experience with Breakthrough to date? This table shows how many we have been granted as of a couple days ago anyway, how many we granted and how many we denied over the course of the program. So you can see that about a quarter of the requests have been granted. The percentage, if I looked at a snapshot in time more recently, would be higher because I think people have gotten a better understanding of what Breakthrough is. So what are some of the reasons for Breakthrough denial? I actually think next time I give this talk, I'm going to say some of the common reasons for Breakthrough acceptance, but right now I'm going to just show this slide. Evidence is too preliminary, and here's some of the reasons. Small sample size, lack of appropriate control, or post hoc analyses of failed studies that identify a subset. And I just want to say with this lack of appropriate control, in some cases, we'd expect to see a control even in the early phase study, and in some cases, the control might be historical. But what is problematic, I think, are the time to event analyses that are trying to make a case on the basis of a couple of months based on comparing to historical data. So that would be difficult to make that case from historical data, but there's other cases where you see an effect that really you would expect that it would not happen in nature. So that might be where a historical control would be appropriate. And then, the subset analysis, it's a problem in this area just like any other. We've had a couple where the improvement over available therapy doesn't appear to be substantial, and sometimes the sponsor hasn't really made a case, hasn't explained it or hasn't made a case for that. And then sometimes the product is modified, so the application will say this didn't work, but we now know we can do this modification and it will work. But that is what we would expect to see some evidence, not the plan for your next development. So I'll talk a little bit more. I know there's questions about what it means to get breakthrough. What are the benefits? Maybe I'll mention that briefly, and I think we'll have some questions during the next panel session. But one of the main benefits, someone gets us, that's different for breakthrough than other expedited programs, is that when a sponsor gets breakthrough designation, we will send them a letter to ask them to submit to us a proposal for a communication plan, and then we'll go back and forth about that. But these communication plans are, I would say, enormously valuable. They set out what milestones the sponsor needs to have certain kinds of discussions. And we've heard from sponsors it helps organize their thinking, like just going through this process. And it certainly helps us in terms of focusing our attention. One of the one of the benefits has been, if a sponsor wants to come in and they really just want to talk about this, and this part of their development plan, and we know that there's this other aspect they're really not paying attention to, it gives us a forum to point that out, which I think has helped a number of our sponsors going through breakthrough. So we get a lot of questions about gene editing, so I just wanted to say a couple words about what we're doing with gene editing. So this is, I don't think I have to tell the audience what is gene editing technology, but it's certainly gotten a lot of attention in the press, and this is how we see it. There's a number of gene editing technologies. This says 4 families, but I'm sure this is, there'll be more families too. But anyway, these are the ones that are reported that we've been either asked about or seen. So the gene editing, what is our approach to gene editing? It's as always a science-based approach to regulation looking at benefit risk. And some of the concerns are the potential to modify the human genome through both targeting and non-targeted effects. And then there's some other concerns that are similar to other things we've already seen with gene therapy, which is unknown long-term effects similar to integrating viral vectors. But I think all of these types of concerns are concerns that we've seen with other types of gene therapy. It's just specifically, scientifically, how to address them that is specific to the actual products. So we've just added to our gene therapy requirements and risks slide that, on-target modification is a requirement and off-target editing is a concern. These are some of the things that we have told sponsors in terms of recommendations to assess safety of gene editing products. So including kinetics of nuclease cleavage and persistence of activity, on and off-target effects, which is what you'd expect as questions. Identification and characterization of off-target effects in cells and tissues, and evaluation of the profiles and types of mutations that are generated. We have a number of efforts to try to look at this from a scientific perspective get outside input. So there's a National Academy of Medicine Consensus Study that this is co-sponsored by OCT GT, but I think it's actually co-sponsored by FDA. And the slide says FDA-sponsored report. It really should say co-sponsored report. It'll focus on advice for the United States and other entities. So it's really not specifically an FDA study and report. I think this slide maybe I could improve next time I use it. But in any case, there's a National Academy of Medicine Consensus Study that is going to look at this area and provide a framework for oversight, and will also give us advice. The kickoff there was an international summit on human gene editing, and then this consensus study was kicked off following that summit. We also participate, as many of you know, in public discussions by the RAC at NIH. They've certainly had a couple of discussions about gene editing. They are noted on this. So I think these have been useful to us and to the field as well. Regulation of human tissue. I've already mentioned the fact that we have a new division director for the Division of Human Tissue. This talk wouldn't be complete without a mention of the meetings that we had. This slide doesn't have it, but anyway, we get many questions, and I'm sure some of you in the audience have asked us some of these questions about whether products can be marketed purely as human tissue or are subject to additional oversight. Our regulations describe the different criteria for being regulated purely as human tissue, but over the years we've become aware that additional clarification of these regulations would be beneficial to the community. So we published 4 draft guidances, and I have them listed here, on the same surgical procedure exception and on minimal manipulation, homologous use of human cells, tissue and cellular and tissue-based products, and also specifically on human cells and tissue products from adipose tissue. We received a number of comments, which one one suggestion we received from a number of organizations was that we have a public meeting. We had a public meeting, which isn't listed here, I see, but we had a public meeting in September to get comments on this. Actually, maybe, so we had a public meeting September 12th and 13th on these 4 draft guidances, and the purpose was to obtain additional comments on the 4 draft guidances. The comment period was reopened for all the guidances together since they had been issued over a period of 2 years, and the issues somewhat relate in the various guidances to each other, so we wanted people to get a chance to comment, seeing all of them together. That comment period closed September 27th. So what are we doing now? We'll be considering information from the public hearing and from the written comments that we received to the docket in finalization of the guidances. And at the meeting, wasn't quite 95, I think that's how many signed up, but about 90 speakers representing a variety of stakeholders, industry, individuals, academic institutions, and other groups who commented to these guidances. So I think that was quite a helpful meeting. I'm going to switch gears a little bit because the Division of Human Tissue also works on things like donor eligibility. And although it is not first in the thoughts of a lot of you in the audience, it does come up, especially when people are talking about international endeavors and cell sourcing or tissue sourcing internationally. So I think it's worth mentioning here. We had these 3 guidances that were published this year that related to donor eligibility, and I only mentioned the hepatitis B one for completion, but I'm going to say a little bit about the Zika virus and West Nile virus guidances because they're a slightly new approach to what we've done in the past with donor screening. So for Zika virus, the guidance, which I'm sure everyone can understand, I don't really need to go into why it's important. The donor screening guidance specifically gives requirements differently for living donors, gestational tissues, donors, and cadaveric donors. And prior guidances have not differentiated between types of donors to this extent. So the evidence supporting these different screening recommendations is that, at least based on information we have at the time the guidance was published, the types of HCTPs with the highest potential for transmission appear to be those recovered from living donors. There's less evidence about the potential for transmission of Zika from tissues recovered from cadaveric donors. And however, and this is noted in the guidance, that understanding of the risks may evolve as more information becomes available, and we certainly could update the guidance document. Well, I don't have one about West Nile virus. I seem to have 2 about Zika virus. I'm sorry about that. But the West Nile virus also, it was issued originally in draft, and then we modified it and reissued it in draft. And it also has some differences for what we have done in the past, in that it has some seasonal testing, and it also distinguishes between living and non-living donors. And the reasons for these distinctions are described in the guidance documents. So I apologize if these slides are going to be posted. I'll be glad to switch that one out before it gets posted. So we have other activities that we do. I just have a brief mention of some of them in this. One is standards activities. So we are very active with standards development organizations and technical committees. We have a standards working group that coordinates our participation in standards. We sponsor workshops or participate in them, and we have some lab programs, including, for example, participation in round-robin testing for standards, and we have collaborations with NIST. But one thing I want to just give a mention to, and I'm sure you've heard other things about it at this meeting, we're very excited about the standards coordinating body that's been stood up or being stood up by ARM, and we're looking forward to working out our participation with that group. We think it's important, and it's important for this community, and it's great that ARM is showing this leadership in standing that activity up. Okay, more standards. So these are some specific examples of committees that we participate in. We also have an active program supporting international regulatory convergence, the International Pharmaceutical Regulator Forum, Cell Therapy and Gene Therapy Working Group. We have a number of activities with in the advanced therapy medicinal area, including the ATMP cluster, participated by FDA, Health Canada, EMA, and Japan PMDA. There's a parallel scientific advice program between EMA and FDA where sponsors can get input simultaneously from the scientific advice working parties and from us on different types of applications. We participate in the APEC Regulatory Harmonization Steering Committee. We've had, over time, personnel exchanges between regulatory agencies, and because of the issues with mitochondrial transfer, we also have an agreement with HFEA. Although at the moment, because legislation prohibits our reviewing applications for certain types of applications, those activities are on hold. Other activities, we do a lot of education outreach guidance documents. I'm going to have a list in a minute of some recent guidance activities, and OCT GT Learn, which is our educational series. We have patient-focused development meetings. These are joint with CBER. Because of our relative small size, CBER has taken the lead on a smaller number of these. But we have been the lead on a couple of these, and I think we have another one scheduled that we'll be the lead on in 2017. We have meetings with patients groups. We make presentations at patient meetings when invited, and we have research activities in a number of areas that relate or are important to patients and developers in regenerative medicine forum and in the regenerative medicine area. And I've included two examples of this here just because they show you the spectrum. On one hand, it's quality of life, which is certainly important when you're thinking about developing treatments, and the other is technical in the gene therapy area. So our OCT GT Learn webinar series, we have added a couple of talks this year and in the process of updating some of these so that they're current. If you're not familiar with this series and you have a question and it's a question where you are willing to sit through, a 20-minute presentation, here are some of the topics, and there's more on this site. People have said it's very helpful. Here's our most recent guidances, aside from the ones in the tissue area. These are some that we have in the cell and gene therapy area, particularly answering questions that sponsors have asked us, like if we have when do we need a shedding study? What should a shedding study look like? Especially since we encourage people to do that early in development. Do we need an environmental assessment? What needs to be in it? So these guidances are in response to questions we've been asked by groups enough that we were ready to write a guidance document in these areas. Here's just an example of the second article that I had listed. There's one of the researchers, Andrew Burns, does research in the adenovirus vector area. Challenges with it include that potential toxicity or difficulties with delivery. So this article that he wrote with his lab, other people in his lab, I should say, looks at the mechanism by which coagulation factor 10 shields adenovirus type 5 from attack by natural antibodies and complement. So doing this kind of research, I mentioned this not because of the specific findings, although they're important and he's a recognized leader in the field, but more just to make the point that it is important for us in these cutting edge areas of science to have reviewers who really understand the science, and having a research component where people are working in these areas helps inform all of us in the office. So last I'll just mention we had a scientific workshop preceding the couple days of comments to our guidance documents, and I'm not going to go into detail here about it. I also didn't put in these slides, but want to mention one other activity, which is we're co-sponsors of the Institute of Medicine, now I think the National Academy of Medicine Forum on Regenerative Medicine with a number of other organizations, and they're having a workshop later this month to look at what can be done to further developments in the field. So I'm going to close here. I have 3 minutes and 23 seconds. I'm watching the timer. So I'd be glad to take 3 minutes and 23 seconds worth of questions.
I have one question on the breakthrough application. There's been mention of an abbreviated form, a 2-page application that is reviewed by the medical division, and to give some sense of whether the review team would think that the product would be relevant for breakthrough status. Has your group have experience with this, and could you comment on that approach as the first thing, as a quote, sanity check, or if you're in the right field to request breakthrough status?
Okay, I'm sorry, I'm having a little trouble because of these lights, and I feel like I'm being interrogated. But I think your question was, are we implementing the 2-page form to look at whether or not it makes sense to apply for breakthrough? Is that what you said?
Yes, and
yes.
Any experience to date about that?
Okay, so I can't tell you at this moment in time whether we already have implemented it, but I know we are implementing it because I've seen people or heard some people in the office talk about how we're implementing it. So whether we're implementing it at this exact moment, or whether we already have been doing it for a month or we're doing it next month, I think the answer is yes, we either are or will be doing it. And the best way to find out what to do would be just to contact the RPM, the Regulatory Project Manager, for that individual file. But I think the answer is either we're doing it now or we're doing it soon.
Okay, thank you.
Any other questions? So I don't know if you're able to comment more about the hearing last month on the stem cell or regenerative medicine therapies and possible new regulations, but my understanding there's a lot of passion on both sides, and maybe some information about how decisions are made for what is done going forward in that process.
Well, you're correct that I can't talk about what we're going to do, but I can comment on the process. So thank you for asking that. As a second part of the question. So this is just what we generally do for guidances, which is we look at all the comments. Some of them are, and even that is going to be a big task with this, because there were quite enough, as you mentioned, there's a lot of interest in this area. So we have a group of people who are sorting through the comments just to get, sort out the duplicate ones. Count them up and put them into categories and see where they affect the different guidances, and then we'll have to go through them all. But because it affects, a number of groups, I'd say it's going to, the office will look at it, Office of Combination Products will look at it, CDRH will look at it too. So there's a lot of interest, and even without all that degree of interest, I think it would require input from multiple groups, just because it does affect them all. So I don't really have a timetable. Obviously, we'll work on this as quickly as possible, but it's going to be challenging to get through all the comments, and really in a thoughtful way, because we want to make sure that we really understand what the issues are. Is there a way we can address them? If we can't address them in the guidance, is there something else we should think about doing that is just apart from the guidances that will be important in development of products? So all of those questions have to be considered while we're going through it. I don't think it's just a question of, okay, we take this comment or we don't take it, but in a thoughtful way, we look at this comment deliberatively and try to consider what it means. What does it mean to sponsors? What should it mean to us? What should we be doing with it to address it? But unfortunately, that doesn't tell you what we're going to do, so can't answer that part.